My name is Emily Fuqua. I'm another PhD student here working at essentially CML, and I'm just going to wrap this session up by telling you guys about a couple of projects that we're really excited to get started in the next, hopefully, few months. The first one is not super sciencey. It's actually a small pilot restoration program funded by the SOAR grant. So this project is entitled Integrating Oyster Farmers into Habitat Restoration in the Florida Panhandle. And the Supporting Oyster Aquaculture and Restoration program, which is funded by the Nature Conservancy and Pew, is a program that really encourages collaboration between restoration and shellfish growers. And so we're really excited to get this money. And we are extremely excited to partner with 5 farms over in Oyster Bay in order to determine if using aquaculture byproduct as an intertidal oyster habitat restoration material is valuable both to restorationists and can also be valuable to the farmers. So we're really excited to get started. Like I said, we're going to focus this program in Oyster Bay. All 5 partners have farms on the Oyster Bay leases. And generally what they'll be doing is they'll be taking their cast off, their unsellable products, their mortalities on the farms. They'll be bagging that up in biodegradable restoration bag material, and they'll be deploying it on our restoration site. The farmers will actually get paid per bag of material that they deploy for us. And we will monitor the restoration as it continues on. And so we're really excited to get started on this. Our restoration site is a series of oyster bars near the leases over in Oyster Bay to make this as convenient to these farmers as possible. So as you can see here, it's kind of a depleted old oyster bar with sporadic oyster clumps. So we know that oysters can still live there, but it needs a little help restoring it back to be a good, healthy, thriving oyster habitat. And so, like I said, the farmers will actually bag the material and deploy it, and then we'll come in, we'll monitor how their sites are developing and how this restoration sort of accumulates into 2026, which is when this grant will end. And so we're really excited to get started. A couple of main objectives of this project are to evaluate the efficacy of using byproducts for restoration, to engage these farmers directly into local restoration and conservation efforts, and to test the biodegradable mulch mesh for use in restoration. So we're going to be using a newer mesh that is actually derived from a starch polymer instead of a biodegradable plastic. The standard in restoration right now is to use a black plastic mulch mesh. And obviously we don't want to be placing plastic into our local systems. So we're excited to also test out a more environmentally sustainable material for restoration as well. Project timeline for this, we are solidifying the contract now with FSU and the Nature Conservancy. And so we're actually hoping to get our farmers started in the next few weeks to get them deploying material and working to get this restoration site up and running. We will be allowing them to deploy material basically until 2026 or until we run out of money to pay them for it. And so we're hoping to get started as soon as possible so they can start getting paid for some of that material. The main project goals are to complete a farmer-led pilot restoration program. We want to create something that could potentially be expanded across all sorts of local places in Florida. We would love to see this project expanded into Alligator Harbor, Indian Lagoon, and wherever oyster restoration needs to take place. And then we're hoping to deploy, we have budgeted the amount of money to deploy up to 2,500 gallons of oyster restoration material before the end of the grant. So we're hoping to make a really big impact on this set of depleted intertidal oyster bars. So like I said, hopefully getting started really soon. And we're really excited to get started. I can't wait. The next project we have coming up is a little more on the sciencey end of things. So it's going to be looking at stress and carryover effects in cultured oysters. Now, before I get into the nitty-gritty, what the heck is a carryover effect and how does it affect you guys, right? So carryover effect is simply when an experience early in life affects how you respond to something later in your life, all right? So I want you to think about what happens if you let your child sort of grow up watching scary movies. Well, most likely your 10-year-old is terrified of the dark, right? I mean, Bing, Paranormal Activity, The Ring, The Shining, whatever, they're scary, right? But later, your 10-year-old gets used to it, right? And so eventually they're not scared of the dark anymore. But now he's a teenager and he brings his friends over and he wants to watch The Shining with them and they've never seen it. What happens then? Well, your kid is fine because he's grown up around scary movies his entire life. Teenagers get sent home traumatized to their poor parents and are now terrified of the dark, right? And so your child has actually undergone what we would call a carryover effect, where growing up around certain stresses changes how they react to it later in their life, all right? So what does this mean for oyster aquaculture? This means that conditions experienced early in life and conditions experienced later in life actually interact with each other, even though they're not experienced at the same time. All right. And so this project is trying to ask questions like, how do larval culture conditions affect long-term grow out of seed in different environments? All right. So do hatchery conditions predisposition seed to perform better in certain environments? And that's one of the questions we're trying to answer. So for this particular aspect, what I will do is I'm actually going to grow larvae in the hatchery, mimicking two different environments. I'm going to mimic the alligator harbor environment and mimic the Oyster Bay environment. And then I'm going to take those larvae through to setting, and I'm going to split those crops, and then I'm going to grow them in those environments, both matched and mismatched. And I'm going to watch how these oysters grow and see how they grow differently depending on whether their early environment and late environment match or they do not match. Another type of carryover effect you can experience actually comes from your parents. So parents can actually change how offspring interact with stress later on. And so I'm also going to complete another project that's asking questions like, how do broodstock conditions affect later offspring performance on lease spaces, and how do they perform in different environments? Do parental environment predisposition seed to perform better? And so I do want to point out that this is not actually a genetic effect. I'm not talking about genes in this case. This is what's called an epigenetic effect. There's a lot of different sort of sciencey mechanisms that I'm not going to go into because I don't understand half of them. But essentially you can have the same broodstock, and if they live in different places, they can actually prepare their offspring better for the places they live in. All right. And so similar to larval carryover effects, only the parents are the ones preparing their offspring. And the setup here is going to be similar. I'm going to have broodstock from the same group. I'm going to condition them and grow them in Alligator Harbor and Oyster Bay, and then I'm going to bring them back to the hatchery. I'm going to grow their offspring out. I'm going to split those crops, and then I'm going to match and mismatch those environments to see how that changes. And so the project objectives for this project are to determine how parental environment and larval culture affect the survival, growth, and conditions of oysters on the farms. So I want to know how those things at the very beginning of those tiny little oyster lives affect their long-term growth and yield on the farm. And then I'm also going to compare carryover effects between triploid and diploid crops. Triploids are well known for having different stress responses than diploid oysters. And so in order to be able to understand some of those underlying differences and understand how you farmers may need to apply triploid versus diploid crops. I'm gonna run this comparison to try to start teasing out what is stress response and what's not, essentially. So the project timeline for this one, I will hopefully be beginning spawns in August. Planting onto leases by October, and I will monitor until about May. So I'm hoping to see market-sized oysters by the end of this project. In Alligator Harbor, I think that's doable. In Oyster Bay, sometimes we'll see. But I'm really excited to get started. If you guys see me in Alligator Harbor, Oyster Bay, this is probably what I'm doing. Feel free to stop and ask. I'm always happy to chat. The main goals for this project, what I hope to get out of this project, are to determine when carryover effects are a significant factor in survival and growth and condition of these oysters. Most likely, there will be certain scenarios when they're really important and some scenarios where maybe they're not so important. And so I'm hoping to start teasing those out. I'm also hoping to create some guides for farmers on if parental environment is important for your leases. How do you select broodstock? Which hatcheries might work with you to create spawns that will work for those specific leases? And how to select seed that may do better on your specific environment lease. And so I'm hoping to contribute to the optimization of Florida shellfish aquaculture by starting to take into account that specific farm environments are just going to need specific things. And I think that is the way a lot of optimization is moving, because what's good for the average farmer may not be good for everybody. And so I'm hoping to contribute to the optimization by taking into environmental specific differences. With that, I'm done, and I would love to answer some questions.
Quick question, which stress markers are you gonna monitor, like heat stress proteins or?
So generally, I'm going to be monitoring just what happens to the overall organism, growth, survival, condition of the animal. So I want to make sure that you guys are growing high quality oysters. If I have the chance, I do a lot of metabolomics for my work. If I have the chance, I would love to run some metabolomics, but that'll depend on funding and things like that. So for those of you who don't know, metabolomics is sort of taking a snapshot of what's going on with the energetics in your body. And so I'm able to look at a lot of specific stress markers like anaerobic metabolism contribution, which is usually a sign of stress, things like osmolarity differences, as well as just what's going on with their general energy reserves.
Probably a better approach than heat shock proteins. A lot of heat shock proteins and never know which one to focus on.
Well, it's a question of, are you looking at a broad set of questions or a very specific set of, you know, so, and I think you're going the right way.
I do too. So this is very interesting. We're seeing two different level approaches for stock improvement. You heard from Heather King and the Gulf of Mexico breeding program, looking at genomics for improvement as well. So a lot of things going on. I'm sorry, there's a lot of questions.
I'll be around. Y'all feel free.